you go.
Do I send you the...
Yeah, if you could share the link to the paper in the chat, that'd be great. Omar, you're still sick?
No. It's only in the morning that I am a little bit like allergic.
Are you still in Bordeaux or are you also back?
No, I'm in Spain now.
Everyone's gone.
In the hot.
Cesare, do you also have the link for it?
Oh, yeah, sure. I'm sorry.
Thank you. No worries.
Need to get it.
Good morning, Chris. He's incognito, not ready for the world yet. Right, it's 9:30, so let's make a start. Welcome to Neuroccino on the 27th of June. This is going to be the last Neuroccino before we take a summer break. But I also want to remind you that we have a CNS talk this Thursday at 4 p.m. Paris time on mapping brain function using high field MRI. And before we get started, I just wanted to share with you this award and say a big thank you to all of you. This is the OHBM Educational Award for 2022. And you all made this happen. You dedicate your time, you present papers, even though you're sometimes uncomfortable about it. So this is really an award to all of us. And I want to say thank you to all for being here and making this happen. Now, briefly, the rules of the game. Anyone is welcome to present a paper they found exciting. Just let me know beforehand so no one is preparing at the same time. And then you just come on and you present the paper and you tell us why you think it's exciting science, and then we discuss it to the best of our knowledge. Now, today we have Cesare present a paper for us, and I'm going to share the link in a minute on YouTube as well. Cesare is currently completing his master's thesis with Michel and me in Bordeaux on a new atlas of the white matter of the human brain. So this is quite exciting work coming out soon. Cesare, if you're ready, and share your screen full screen, please.
Absolutely. You see it?
Yes, fantastic.
Good.
Take it away.
Okay, thank you, Stephanie. Yeah, today I'm presenting mapping microglia and astrocyte activation in vivo using diffusion MRI from a group at the Neuroscience Institute in Alicante and Cardiff too. And it's a pretty cool paper actually, because well, we know that glia is involved in many neurodegenerative diseases, Parkinson's disease, Alzheimer's disease, MS. And all these diseases are characterized by chronic inflammation that leads to neurodegeneration. But we don't have good measures to have a look at the glia, which is mainly involved in this inflammation and therefore neurodegeneration. We have PET, for example. That's the silver standard we have so far. PET implies radioactive tracers, and this means that we cannot use it for longitudinal studies, for example, and clinicians have to be careful when using it with elderly patients, which are the most vulnerable population we have. And those are the ones that are mostly affected by neurodegenerative diseases. So we have a problem here. Now, conventional MRI is sensitive to morphological changes caused by neuroinflammation, but it cannot differentiate between subpopulations of the glia. The authors here proposed a new imaging framework that is based on diffusion-weighted MRI that allows to image both microglia and astrocyte activation in the gray matter non-invasively. And they also demonstrate the translational validity of this work that has been mainly done on rats in humans. It's an important work because it could lead to the characterization of the tissue inflammatory state. And so they could provide with this, and future studies could provide with this early disease biomarkers or prognostic ones. And this would definitely be very useful when facing neurodegenerative pathologies. First things first, how do they do it? How is their MRI protocol? Well, basically, they built this tissue model of gray matter diffusivity that accounts for microglia and astrocytes morphology. I don't want to go too much into the details here, but basically what we have is a model that accounts for the water diffusion of these two subpopulations of glia. And microglia and astrocytes are modeled in two distinct ways. So for the microglia, they identified 3 MRI parameters, which are stick fractions, stick dispersion, and small sphere size. We'll get back to them later. And for astrocytes, we have only one MRI parameter, which is the large sphere size. So to give you an idea, this was the modeling of microglial cells with the small sphere and the sticks accounting for the cell processes. And this is what they identified for the astrocytes, a larger sphere, basically. To validate the model, they had to perform 3 experiments, 3 different experiments. So first, they dissected the different MRI signatures of microglia and astrocytes activation. To do that, they injected the dentate gyrus of rats, only one hemisphere, with LPS. LPS is lipopolysaccharide. It causes an acute inflammatory response because it triggers the release of many cytokines, many different types of cytokines. And with this, they could dissociate the response of microglia and astrocytes because these two subpopulations have different activation windows. So microglia gets really active already 8 hours post injections and even 24 hours after it. Astrocytes instead starts to be really activated only 24 hours post injection. So here you see a normal microglial cell, and this is 8 and 24 hours post injection. You see we have a bigger soma and less cellular processes. This is reflected, this implicates microglia activation, so inflammation, and it is reflected in the MRI. We have decreased stick fraction. Now, stick fraction is the amount of water that undergoes restricted diffusion. So it's basically a measure of the degeneration of the processes, meaning that if we are losing stick fraction, we are losing the microglial processes. And you see here at 8 and 24 hours, we have a marked decrease. And we also have an increase in the sphere radius, reflecting the increase in the soma size of microglia. Now, this allowed to image the microglia MRI signature. And they did the same thing with astrocytes. as I said, they were able to temporarily dissociate them, basically. And because astrocytes show an enlargement after 24 hours, they went to look for that. And in the MRI, we have an increased sphere radius exactly after 24 hours. Now, this finding is particularly clear because they injected the rats when they were looking for astrocytic activation. They injected the rats with the CSF1R inhibitor, which depletes about 90% of microglia, so they remain only with astrocytes, basically. And with that and the different time window activations of the two populations, they showed this nice finding. So here we have a condition where we have neuroinflammation, but no neurodegeneration. This allowed to disentangle microglia and astrocytic activation, but they wanted to see if this work was going to be replicated in the presence of neurodegeneration. So they performed a second experiment to see basically if the model could differentiate glia activation and glia proliferation. Proliferation is an indirect measure of neurodegeneration because it's associated with chronic inflammation and through that with neurodegeneration. And to do this experiment was particularly important because it shows that the model can inform us about the inflammatory status of the tissue in ongoing neurodegenerative pathologies. And because that's the clinical reality, let's say, it was critical to show this. So they injected rats with ibotenic acid to cause neuronal loss. We see here in figure B, we have a severe neuronal loss in the rat's hippocampus, and the histological analysis showed this, but also it showed a distant microglial response, although no astrocytic activation. These same findings are again mirrored in the MRI. We have a decreased tick fraction, increased dispersion, and very importantly, an increased small sphere fraction. This is a signature of microglia proliferation. And this means that with this parameter and the other ones specific for the activation of microglia, they were able to differentiate proliferation and therefore neurodegeneration from inflammation. And this means that the model is still valid in the neurodegenerative condition, let's say. The third experiment they did was on demyelination, because demyelination that happens in MS, for example, can alter greatly the water diffusivity of the tissue, and that could have been a confounding factor for the model. So they demyelinated the rat's hippocampus in one hemisphere, and what they found was that one of the three MRI parameters for microglia was sensitive to demyelination, and that was tick fraction, but the other two were still good. And with them, they could discriminate between demyelination and microglia and astrocytic activation. So demyelination is not really a problem with these findings. It's still good. The model is still good, and it's quite stable. Finally, they wanted to prove translational validity of this work, because this is all done on rats. And so they scanned 6 subjects with a 3T MRI and found 2 things. First, the model correlated well with the microglia density maps that were created from postmortem studies. So microglia is positioned in different densities across gray matter regions. And they found that the model could predict that compared to the postmortem histological studies. So this means that the model is useful to generate glia biomarkers in the first place. And second, they showed both within and between subjects coefficient of variations that are in the range of conventional MRI. Conventional MRI used in the normal clinical practice. And so the glia biomarkers that this framework can provide are highly reproducible. If we look at these findings, obviously 6 subjects, it's not much yet, but still, these biomarkers are reproducible between and within a cross-section subjects. Yes, I think this was a really good job from them.
Great. Thanks for presenting this paper, Tesile. Are there questions either here in the room or over on YouTube? If you have questions, just write them in the chat and I'll bring them over here. If not, I'm going to go first. Very well done. Thank you for presenting it. A couple of questions I have. The first one is that you showed the validation of the model in the rodents, and they injected on one side, but not the other, and then compared the 2 hemispheres. Here we go, exactly. Is there no spillover? As in, does it really stay in one side, or does the injection travel, or is it a matter of timing?
I don't know, because they didn't mention it. I don't know it on my own. I went for the supplementary materials, but you have to ask for them, so didn't get them.
Okay.
Yeah, I can't really answer to your question. I guess they specifically see 2.5 micrograms to microliter is shown here. I think they calculated quite well the quantity of LPS in this case, but in general of all the substances they injected, and probably they didn't have spillovers. I mean, it was a really focal injection. It was not even the hippocampus generally, it was the dentate areas of the hippocampus. So I'm expecting super small quantities, very well quantified.
Great, thank you. Chris has a question.
Yeah, thank you for the presentation. So it's DWI, but is it, well, I'm guessing it's a different sequence. It's their custom sequence, right? Do they?
Yeah.
But, yeah, so it's not possible to use existing data to do what they do, right?
Well, they adapted the model to humans to do the validation, the translational validity studies, and I've seen in the supplementary materials that I didn't actually see, but yeah, the website, they basically they ask you to ask them for the pipeline. If you want it. So maybe gonna be useful for that? I don't know that either.
Okay, thank you.
Chris, I just scrolled through the paper and there's a section saying comparison with conventional MR techniques and translation to human. So you might actually find some answers
Cool.
in there. I'm guessing, given that Derek Jones, I think, was amongst the authors, this should be fairly applicable to other scanners.
Thank you.
Any other questions here? If not, I continue for a little bit. So more a, gosh, I'm doing this annoying thing, more a comment than a question. But when we talk about humans, it'd be great if we not call them subjects. I know it's in many papers, but it's a constant battle of mine. It's participants, it's volunteers. Call them anything, but not subjects.
Yeah, I know you have an issue with that word.
It's not just you. I just wanted to put it on record because it's in many publications still, and I think we've been having this debate for, I think, as long as I'm around. And I don't mean me in science, but me on this planet. And it's time to just move away from calling people subjects. So that was my annoying hat on. I have a comment, not a question, and then I have an actual question for you. And you're actually showing the figure at the moment. So figure 6, panel B. If I understood you correctly, they show the correlation between postmortem and the diffusion measurement in terms of the, what was it? The microglia density. And thanks. And I'm guessing we see a confidence interval here of some sort. So are there parts of the brain where the model applies better than other parts? The occipital lobe, for example, are these outliers? Or
no, I don't think they are outliers. I just think they fit less in the progression. I mean, you see some of them are pretty clear here. And well, a limitation here is that they consider only 8, if I remember correctly, 8 brain regions. So it's not like a whole brain thing. Some of them, but I mean, if you take out the occipital lobe, you have a really nice correlation here.
That's not how statistics work. You don't just take out the part to make it look.
But I don't think that the occipital was an outlier. They checked for it and they didn't say anything about getting rid of them. So it's part of it, definitely.
Okay. Ma has a question as well.
Hello. Yeah, I wanted to ask, because I suppose these participants that they use for validate, they are like healthy controls.
Yeah.
So my question is, what do you think, or if the authors discuss this in the discussion, how far or how close are we of applying this method to brains that are of some pathologies, or with this, when this can be like interesting?
Well, I think that before this to be like something really useful in clinical practice, we need some more time. But it's really, I don't know, interesting, and I mean, gives us hope, I think. And I think it probably would need more validation on humans first, but after that we can move on to pathological cases. I mean, it's designed for that, so I don't see it as something that has to come like in 10 years. Actually, it would be really helpful. Then they stated that too. It would be really helpful to try the model on like an Alzheimer's patient. Yeah, definitely, it's created for that.
And I guess given that it's diffusion based, non-invasive, it should be fairly quickly translatable into in vivo studies and pathological cohorts, right?
Yeah, exactly. I mean, as I said in the beginning, it's not path, it's diffusion. So non-invasive, 100%.
Did they specify anything about the type of scanner that you can use it with? Do you need to have one of those fancy new scanners, or can you?
No, it was a 3T here, a human 3T connectome scanner. Like, no expert on scanners, but
2 millimeter. Okay.
yeah. So it's also quite high resolution.
So it should be applicable fairly soon, hopefully. Right. I don't see any other questions. In that case, thank you very much, Cesar. Great paper. Thank you all for being here today. And then a quick reminder again, we're taking a summer holiday starting in July and half of August, but we will be back this Thursday with a talk on mapping brain function with ultra high field MRI by Dr. Witzke von Litzwag. So do come and join us this Thursday at 4 p.m. Paris time. And other than that, I wish you an exciting week in science. Thanks for being here, and I see you on Thursday. Bye.